Welcome everyone to the Herbert Wertheim College of Medicine Journal Club on all things COVID-19 pandemic. We are back and better than ever. I hope everyone has realized the value of our P3 app and enjoying the repopulation efforts here on campus, considering the social distancing and mask wearing measures that we all share. And hopefully you can enjoy that in-person meeting with your students and coworkers. My name is Chuck Dimitrov, the Executive Associate Dean for Research at HWCOM. And as we enter our 49th week, we are here to educate you in the form of a journal club on everything COVID-19 pandemic. We are at the precipice of this pandemic, ready to turn the corner with the rollout of new therapeutics and sophisticated new types of vaccines against this insidious SARS-CoV-2 disease. Today's journal club is led by Dr. Kalai Mathee, a professor and infectious disease expert in the Department of Human and Molecular Genetics at HWCOM, with Dr. Andrea Raymond, an associate professor and immunologist in the Department of Immunology and Nanomedicine, along with an HWCOM MD student, Kevin Bennett. We are also really glad to have and excited to have a special guest, Dr. Lindsey Baden, a renowned clinical investigator and viral disease expert from Brigham and Women's Hospital, Dana-Farber Cancer Institute. Dr. Baden, in fact, serves as the principal investigator of Moderna's COVID-19 vaccine clinical trial in the Boston area. We are lucky to have him in attendance today, and he certainly will provide clarity to any outstanding questions or will provide more in-depth analysis. The topics covered today in today's presentations include a historical perspective of vaccines, how an immune response is generated to a vaccine, and thirdly, describing the safety and efficacy data on the major COVID-19 vaccine candidates. Because of the sheer expediency, 9 months rather than a normal 5-year time span, and the bureaucratic exuberance to which these vaccines were mass produced, in conjunction with Operation Warp Speed, I sense a reluctance and tepid enthusiasm for their buy-in from our community. However, I can assure you that the rigor for evaluating safety and efficacy was imposed in these trials. We hope to provide you with the necessary details and confidence to jump into this country's efforts to vaccinate everyone and build widespread immunity. And right now, I'd like to turn it over to Dr. Mathee.
Thank you, Dean Dimitrov, for the nice introduction. I'm very excited to lead this discussion today. Just some disclaimers so that, perhaps I know a little more than you do. Perhaps, many of you know more than I do. So please feel free to answer questions that you think you can answer on the chat. I want to remind that I'm not a medical doctor. Often I get questions asked, but please consult a physician. Having said that, our panel member, Dr. Lindsey Baden, is a medical doctor. You can pose questions for him. Though when we were starting to prepare this journal club, I realized that I was very ambitious, that I wanted to cover at least 5 vaccines. And as we started preparing, realized that we only have time to cover Moderna and Pfizer vaccine, which are currently approved for emergency use. And certainly in January, when AstraZeneca goes for approval, we can certainly have another session to discuss how those vaccines were constructed and what is the outcome of their clinical trials and their tribulations. So we will, the first part is obviously Kevin and I going to lead the discussion. We roughly will talk about the smallpox and variolation, because you can't talk about vaccine without talking about smallpox. A very brief, layman's overview of vaccine and immune response. And then I'll get into a little bit about types of vaccines and those are in use, and give you an overview of what it requires in preclinical and clinical research. Then we get into SARS-CoV-2 vaccine. I'll turn over the presentation to Kevin to talk about the Moderna and Pfizer vaccines. And then I'll come back to discuss why there are so few, why is that even we are so fast. And then we'll move into the panel discussion. So variolation is a process where they were introducing viruses in, did I move? Okay, before I go there. So smallpox, many of all of us are is vaccinated, and most of us, if not, and it is eradicated for as of now. There are some appearance of other pox in the world. It is a devastating and a disfiguring disease, and it has about 30 to 40% fatality. How you get infected is either by contact or inhaling. And it's caused by a virus, a DNA virus. Its nucleic acid is DNA and is known as variola. There's absolutely no cure for this virus except for prevention. And as early as 1000 AD in the Chinese literature, we find that there is a process called where they introduce the scabs from patients with smallpox into the nostrils of healthy individuals, as shown here. And there's also indication that variolation process was already in place in India in 1500. And then in Africa in 1700s, when the Boston doctor found that slaves had this variolation where they scratched the skin and introduction of the scabs. And then the first most famous story is that when Lady Montagu was in Turkey with her ambassador husband who got the smallpox, and she heard about variolation and she variolated her son. She brought that process to UK in 1721 and variolated in public her daughter and quite a few people. And they did experience 2 to 3% fatality. And remember, if you don't have variolation, there was 20 to 30% fatality without that. The first time a variolation was done in the US is in 1738 in Charleston, South Carolina. About 445 people were variolated with the 4% fatality. Just like now, all the politicians are up there in public getting variolated. So were the leaders those days, like the Chinese skiing or the Catherine the Great, some of the examples, there's many more. We don't have time to go through. And then, of course, everybody knows about the story on Edward Jenner, who was born in 1749, where he actually introduced the process called vaccination. And how did he get started? He heard the stories how women who are milking the cows, so those days they milked with their hands, and they often are very immune to smallpox. They have little sores from the cowpox. And then he argued that maybe something in this cowpox protects these individuals. So he took the fluid out of cowpox from a nurse and introduced it from a milkmaid, introduced it into James Phibbs, an 8-year-old boy, and he found that when he challenged a boy with smallpox and cowpox, and this boy did not get sick. So they call the process vaccination, and he credits the word to Richard Dunning, her fellow physician. And the word vaccinations comes from vacca, for Latin for cow. And when he tried to publish it, they ridiculed him. The church called it ungodly and repulsive. And the Royal Society rejected the paper and warned him not to publish anywhere. But he published it anyway in 1798 with additional experiments. And then in 1840, there was free vaccination to all infants, and UK was instituted. Since Jenner, we have many more vaccines. It took almost another 100 years. We got the second vaccine to rabies. And then the golden era of vaccine development is really 20th century, rather mid-1950s, where we saw many, many vaccines to come online. And why does it take so long? Because a good vaccine needs to be able to induce immune response without a disease. So that means our body generates the antibodies so that they can neutralize or combat the incoming pathogen. It should be non-toxic, no side effects, non-adverse reactions. It should provide lifelong immunity. As you may already know, that's no longer true. We do need booster vaccine as you age, especially if you're over 55. And then the thing is, in order to figure out what should be a good vaccine, you need to think about what does our host immune system see when a pathogen enters. So when a pathogen, whether it's a virus or bacteria or parasite, it only can see what is on the surface. Usually these surfaces of these pathogens have proteins. They make a good immunogens. There are a couple of bacteria and some fungi that have sugar molecules coated on them. They are known as the polysaccharides. They make good conjugate vaccines. Again, I don't have time to get into that. Some other things that the host system sees are the products that produced by pathogens such as toxins. Of course, you don't give the toxins per se, you inactivate the toxins and give them as vaccines. In terms of the immune response, and I just want to use this slide as an example to show you, when you have the vaccine introduced into your muscles through the intramuscular injection, the first set of immune cells, which I call them the foot soldiers, they are circulating in your blood vessels and your lymphatic system. They see this presence of this foreign entity, they engulf it, and then they will move or take those component to the host lymphatic nodes, where the lymphatic nodes, they inform the T cells and the T cells particularly to say, hey, we have an invading or we have some foreign material. We need to send our cavalry and we need to send to the site of infection, so to speak. And then they will actually, have the T cells differentiate to cytotoxic T cells. In the case of viral infections, they will migrate to where the infection is to try to get rid of all those cells that are infected by the virus. And then they also inform the B cells, which then differentiates into plasma cells. The plasma cells are essentially the factories that make the antibodies. And these antibodies then also go and help the T cells finish the job of clearing the pathogen. But what is very important is that in this process, they also create what we call the memory cells. These memory cells are important. They get filed away, and they're called upon to action as soon as this pathogen shows up anytime in our lifetime, most of our lifetime. So, and then there are specific chemicals that are very hallmark or a signature chemicals are made during the process. The interferon gamma is very important for viral infections, and IL-4 for making the antibodies. And I want to talk about why how our immune system responds as soon as we have the pathogen or the initial exposure to vaccine. We have what we call a primary immune response, and this happens within 10 to 14 days. You start seeing the antibody to 3 to 5 days. You start seeing the antibodies in our system, and this is the signature antibody made at this initial immune response is an antibody which is IgM. And then when you give the second shot, and what happens is you have the secondary immune response, which is much faster. The memory cells immediately react very quickly. And not only it's faster, it also goes very high. At this point, we are making these IgG antibodies, and these are the critical antibodies. These are what we call also the neutralizing antibodies, and these are very specific for the pathogen. So then they can go and recognize the virus specifically and neutralize the virus. So there are several kinds of vaccines. There are 5 types of vaccines. The early vaccines that were introduced were the attenuated viruses and bacteria, where you passage the pathogen in the lab. When you grow them in the plate, when you're not away from the humans, they change. They don't need to make all the products that make you sick when they're growing on the plate. So over time, they lose that function and they can still grow, but they don't cause the disease. So many of our childhood vaccines are still these attenuated viruses. And of course, you see BCG is an attenuated bacteria, which was introduced in 1930. It's being continued to be passaged. And then came the inactivated vaccine. The scientists discovered that you don't have to give any live pathogens or live bacteria or virus. We can just inactivate them wholesale and give them the total inactivated component, and then we can get the same protection without any disease. And we, of course, have a bunch of them here listed. And then came the subunit vaccine. So instead of just giving everything in the pathogen, let's figure out, what is the molecule that is on the surface? What is the molecule that causes disease? So that's when they ended up generating a vaccine very specific for the molecule or the protein molecule on the surface. The first subunit vaccine introduced was hepatitis B subunit vaccine for the surface surface molecule, surface Hep B protein. And of course, toxoids came here, and of course, polysaccharides, which were important for preventing meningitis in people. And then came the 21st century. 21st century brought in new methods to prepare vaccines. The first recombinant vaccine, and this time what they said is, let's put a piece of the subunit information into a live virus, which doesn't cause disease or cause like little cold-like symptoms, such as the adenovirus. If we integrate that piece, now the virus will replicate, it will make this protein, then we'll develop vaccine. So the first and only approved recombinant vector vaccine is the Ebola vaccine, and the phase 4 trial is now ongoing, that started in December 2019 when it was approved. And of course, now 21st century, we are in 2020, we have this mRNA vaccine. We're going to talk about that a little bit more in length. What does it take to go from bench to population? Immediately, first you have to do the discovery. You find the pathogen, you find what causing the disease. Once you identify that, you got to do the preclinical testing, then request for permission to do the human trials. Once the human trial starts, that's the clinical research, and then you apply for FDA approval in this country. Different countries have their own approval agencies or bodies, and then they analyze the data and they give the approval or give them the license. Only then they may start the large-scale production and the phase 4 trial, as well as continue to follow up the individuals that were included in the clinical research. And as you can see here, what is this clinical research entail? Oh, before I go there, in the past, everything was done sequentially. The discovery alone can take 2 to 5 years. Then preclinical testing can take 2 years. And then the clinical research can go up to 5 years. And so usually it gets up to 10 to 15 years before there is a full approval to use a vaccine mass scale. What happened with COVID-19? We did not have time to do the sequential. So everything was done simultaneously, or most of the work was done simultaneously in anticipation of having a vaccine ready to be deployed. So in the preclinical testing, once you do the laboratory test, isolate the virus, and then again, technology played a huge role. We were able to sequence the virus in a day or half a day, essentially, and analyze it, and understand the host immune system. That's part of the discovery process. And then we had another major component at our fingertips, the bioinformatic analysis that allowed us to analyze the data, millions of base pairs in no time. And then we also had the high performance computing opportunities to use the high performance computing to identify targets in a much faster. What could have taken 2 weeks, 3 weeks, we literally can do 1 hour, especially with the cloud computing, we can move forward very quickly. And then this then takes us to the laboratory test. You express the vaccine, you check the stability, you figure out what is the formulation you want. But even then you have to do the animal test to see that this vaccine actually works. How do the animals respond? Whether these vaccines can protect the animals, which we call the challenge experiments, which of course we can't do in humans easily. Having said that, there are ongoing challenge experiments that has already been started for some of the vaccines. So again, these are, in the past, we do it sequentially. With COVID, we do everything simultaneously. That's only possible if there's plenty of money in the process. So the clinical human trials, as you can see, there are major 4 phases. And once they're approved for testing in humans, the phase 1 is really to determine whether this particular formulation is safe. You use fewer participants, you ask only healthy people, and you identify whether they're safe or not. And then you do the phase 2 where you determine the dose you need to give, and you also include some patients. And then the phase 3 clinical trial, which I heard a lot about, is the safety and efficacy portion, where you actually do a double blind. The people don't know what they're getting, neither do the providers know what they're getting. They're placebo controlled because there's a group that gets the vaccine, the other group that gets the placebo. Of course, you don't know which one you're getting. You involve many more participants. They recommend 300 to 3,000 participants. And then it's submitted for approval. And but 800-page documents is submitted to FDA. FDA paws through the data. The biostatisticians play a huge role here. They then distill the information to 40 pages or 45 pages for us to review, and FDA also distills it to 6 pages for the health community, the vaccine providers. And at this point, and once it's approved, that goes for phase 4 trial. And as you can see, they can ask for emergency use of these vaccines. And some of the vaccines, like the Russian vaccine, went into emergency use soon after soon after the phase 2 trial, even before completing phase 3 trial. So these are the 4 major phases of the clinical trials. And that brings us to SARS-CoV-2 vaccine. As I told you earlier, once we know the pathogen, we isolate the pathogen, we photograph it, doing EMs to identify what it looks like, and then determine what is the genome look like. And of course, SARS-CoV-2 virus is a coronavirus. You can see the corona, the tiara around the virus, and then it contains an RNA genome, and I don't have time to discuss this. If there's a question, I'll address that later, the implications of having an RNA genome. But what you see on the surface is this red color proteins, and that now we know these proteins are known as the spike protein, and the spike protein is very important.
But make sure you tell everybody that spike protein is a spike glycoprotein, and all that beautiful orange stuff is actually glycans.
Absolutely, so, sorry. Okay, so this, like Dr. Dean Saxton pointed out, this spike protein is a glycoprotein. It's heavily glycosylated, which means it's covered with the sugar molecules. And that actually also protects the virus from immune system to some point, because our cells are also our proteins on our membrane as cells are also heavily glycosylated. So the spike protein, why is it important? When they enter our host cell, our cells, they actually make a direct contact with our receptor protein. And we now know that the receptor protein is the ACE2 receptor. So the spike protein has to make this contact in order to enter the cell. So this is why it's actually a very important vaccine target, because this is the first, and if you have an antibody that will cover this spike protein, then you get protected from any infection. And so from there we go. So what does the landscape of the vaccine look like, a vaccine development? As of December 2020, there are 184 vaccines in development and 89 in human trials. We have all kinds of vaccines, protein subunit to live attenuated to inactivated virus, recombinant vectored virus, as well as, of course, we know nucleic acid based. There are 37 of them, and 10 are in clinical trials. And of course, we know of 2. And what does the landscape look like in the vaccine trials? 44 has entered the phase 1, 19 has entered phase 2, 18 phase 3, 5 have been approved for limited use. That is those Russian vaccine and the Chinese vaccine, and among the others. And of course, 2 have been approved. That is your Moderna and Pfizer vaccine. And we have 1 vaccine already abandoned, which is the Australian vaccine. Again, if you have questions, we can talk about it later. Why was it abandoned? And then if you look at these vaccines, the leading vaccines, you can see that there are all kinds of vaccines that are leading vaccines. The 2 are the Moderna and Pfizer vaccines. The type of vaccines is the messenger RNA. Both of them have been approved now by FDA. But what is important to note is it's a nucleic acid vaccine, it's an RNA vaccine. And in terms of nucleic acid-based vaccines, the technology is not new. People have been working on it for more than a decade now, try to figure out how to introduce a DNA vaccine. And there are concerns, and again, I don't have time to go into the details, but important to note is that technology was already there, and there are 3 vaccines are being evaluated as we speak. And then comes along the RNA vaccine, which is the SARS-CoV-2 vaccine. So no DNA vaccines have been approved, and now we have 2 RNA vaccines that have been approved. And these 2 RNA vaccines, of course, target the spike protein. This Pfizer vaccine, which is a US-Swedish company, combined with the BioNTech, the German company, to create this vaccine. They use the full-length spike mRNA. That means they use the entire sequence of the gene, spike gene. Whereas the Moderna vaccine, which is a US-based company, they actually used only the receptor binding site, and they used 3 copies of the receptor binding site, so it's a trimer. And this is the major difference in terms of the message. So here's a full length, and whereas here is a trimer. And what I'm going to do now is, this is a very short 1-minute video, which really shows us how this vaccine work. And while I show this video, and then we'll come back to, okay.
It all started with isolation of the virus from an infected patient in China. The coronavirus was sequenced, and its genetic information was uploaded to a public database. Working together, scientists at the NIH and Moderna identified the sequence for a key protein on the surface of the virus, called the spike protein, as a good vaccine candidate. The instructions for making the spike protein were then encoded into an instruction molecule called mRNA, which could be administered directly to patients as a vaccine. The vaccine is injected just like other vaccines. The mRNA is taken to immune cells, where it instructs cells to make copies of the spike protein, as if the cells had been infected by the coronavirus. Other immune cells, shown here as red and orange figures, are then able to learn about the spike protein and develop ways to protect the person if they ever come into contact with the actual coronavirus. What makes this approach different is that you don't need to make the virus itself to make a vaccine, a time-consuming and intensive process. Instead, you use the information from the virus and administer the information directly to the patient. Essentially, the patient makes their own vaccine. This cuts out the middleman. Using this approach, NIH and Moderna were able to go from sequence to a vaccine ready for human testing in record time, just 42 days.
So that's a very nice video, I thought, that allowed us to understand a little bit more. And here we move to now Kevin, who will just present the clinical trials. Go ahead, Kevin.
All right, thank you very much, Dr. Mathee. Okay, so here we're looking at the vaccine formulation, so all the ingredients that are going into the actual vaccine that we're going to put into people. So the 3 main components of the vaccine are the mRNA, which is the thing actually encoding the protein that we're trying to develop the immunity to. We also have the lipids, which form a ball around the RNA to kind of mimic our own cell membranes and to protect the mRNA from the environment outside, and to allow for effective delivery of the mRNA into cells. And additionally, there are various other substances that are added primarily to stabilize the lipid membrane or maintain a specific environment around the mRNA inside of the lipid nanoparticle to keep the mRNA from degrading before its job's done. So these ingredients are particularly important because if there were some unintended adverse reaction or side effect to the vaccine, it may very well be from one of these components and not necessarily from the immunogenic substance. Next slide, Dr. Mathee.
Oops. Okay.
Okay, so as I mentioned in the previous slide, these vaccines require a lot of attention to ensure their stability. As you can see here, temperature plays a big role in the stability of the little lipid nanoparticles. And a particular note, Moderna's vaccine is stable at room temperature for up to 12 hours, This is really important for practical use so that you can transport the vaccine without an ice chest or a deep freezer, or keep it out in vaccination centers all day long, not worry about it losing its potency. Next slide. Okay, so the vaccine delivered in an intramuscular injection, just like you'd think of with many other vaccines. And on the side here, on the right side, there's a good representation of what we saw in the video of the mRNA being wrapped in the lipid nanoparticle, delivered into one of our cells through the injection, making the viral protein from our own machinery, and then allowing our immune system to kind of go to work on it. And during the Moderna and Pfizer trials, they provided 2 doses of the vaccine that were timed slightly differently between the 2 of them, but nonetheless, they were timed to optimize that process on the previous slide. So we can go to this one now. So these are some of the numbers, and there's going to be a lot of numbers. So just hang on through these next couple of slides, and we'll make sure that you understand them. This table is showing the evolution of the Moderna clinical trial and how they came to settle on the parameters for their phase 3 trial, which is what we're all here to learn about. So under Moderna, you can see that as the trials progressed, the numbers of participants increased, like Dr. Mathee was saying. When you go from phase 1 to phase 3, we're kind of changing what we're looking for, so we need to add more participants. Until they finally found a dose that was safe and showed a good immune response. And next slide. Yes, so here with Pfizer, although it's slightly different because they combined their phase 1 and 2 and 2 and 3 trial, you could still see that that they increased participants. They finally found that dose at 30 micrograms, and they additionally had the 2-dose schedule, which theirs was a little different than Moderna, but a 2-dose nonetheless. Also, that the trials were controlled with normal saline is good to note. Next slide.
Yeah. Oops, sorry.
This one, yes. Okay, so the phase 3 or the phase 2/3 in Pfizer's case, clinical trials were pretty quick when you think about the short time span of the pandemic. Pfizer's has an earlier start date, but this is likely because of the combined trial with phase 2 and 3. But it's noted here that both groups finished collecting data to send to the FDA for approval a month ago. And now we have people receiving the vaccine, which is extremely quick. The vaccine protocols they settled on can be seen here with the vaccine dose, the placebo used, the number of shots given. And another important point in differentiating the 2 trials was that we see here that Moderna had 100 sites that were confined to the United States. However, Pfizer had a larger number of sites as well as an international presence. This will make a little bit more sense. We can clear up some of the issue here when we go into the demographics in just a moment. Next slide. Okay, so the demographics of Moderna phase 3 were a slight male predominance, and only adults recruited in the trial, which should be followed by saying that there is a Moderna trial that is currently recruiting adolescents. However, that information isn't included here. We also see a predominance of white participants. It's all well matched in the placebo group as well. And we see on the Pfizer side that the participants were not too dissimilar. Pfizer has a larger population in general, but we see a very slight male predominance, and that Pfizer notably included participants from age 12. So the trial that Moderna is currently doing, Pfizer included in this trial. Okay, so our outcomes. We start with the schedule. Both groups had their participants come back to the testing site kind of serially to have their blood drawn to test for COVID, to ask about symptoms or any bad reactions that they may have had. So both groups tested for COVID. Both tested antibody production, with Moderna also testing for the ability of the antibodies in the patient's blood sample to neutralize the virus in a lab with the neutralizing antibodies there. Moderna also tested for cytokine production, which is kind of a broad indicator of immune response. However, the profile of that cytokine response can inform the researcher on if what they expect to be happening in the body is actually happening. Additionally, they made sure there weren't any serious adverse reactions occurring, and they kept a log of any side effects reported by the participants. Next slide. Okay, so quickly, this is a side-by-side comparison of some common side effects that are seen with vaccine administration. And the important thing to know from this slide is that most vaccines are going to come with some unwanted side effects like pain or fever or just flu-like symptoms. But a good vaccine is not going to have any serious health concerns, the serious adverse reactions, which is what we saw with these two vaccines. The next slide. Okay, so graphically, we can kind of gain a good appreciation for just how innocuous these vaccines really were, especially when you compare it to actually getting coronavirus. It's quite innocuous. So on the Moderna side, if you look at the left column, the far left column, this is those who received the vaccine, and in that green part there is the percentage of those with no reported side effects. So it's about 94% or so of the group. The remainder is those reporting injection site pain or a generalized reaction of some combination of flu-like symptoms. And then directly to the right of that in the middle is the placebo group with almost the exact same numbers. And this is good because it means that the contents of the vaccine are probably not dangerous, and that a good percentage of the effects are potentially psychosomatic or just not related to receiving the actual vaccine, those lipid nanoparticles.
Can I correct you, Kevin? Remember,
sure.
they were given saline as the placebo, so they didn't give the nanoparticle without the mRNA.
Right,
so
right. I might have kind of not been totally clear on that, but yes, they did get just saline. And then so on the right of that is the total, the combined of the two, and as you would expect, the numbers are somewhere right in between. Then we look at the Pfizer numbers, and while they are somewhat the same, we see significantly more reaction in the Pfizer participants who received the vaccine. But however, 70% of that group did not have any side effects. We see the placebo group to the right was approximately the same as it was for Moderna, and overall, it was still a very, very safe, very efficacious, or rather a low side effect profile, is what I'm trying to say, of about 80% of people did not report any side effects. Next slide. Okay, so now for the efficacy. When you compare the efficacy of the vaccine to the efficacy of the placebo, which again is just salt water, and you look at how many people in each group contracted COVID, we see that the vaccine group was far more efficacious. You can think about the numbers under efficacy as the value of the vaccine versus the value of getting the placebo. And if you look at all participants and then the percentages that are associated with them, this is the real efficacy that we're looking at with these vaccines at 94 and 95%. So it's pretty good. And then the next slide, Dr. Mathee, or the next click. Here we go. So this is the efficacy of the vaccines when you split it between male and female. And while it is still excellent for both, it's important to note that there was a measurably lower efficacy in women across both groups, the Moderna and the Pfizer. So the participants in the placebo and the vaccine groups were not given an injection and then exposed to the virus, just to kind of harp on this again, what Dr. Mathee said. Rather, they were kind of given the injection and sent out into the world at equal risk of contracting the virus. They did make sure in both studies to also include high-risk populations. For example, my godfather is a doctor and his husband works in healthcare, and they both participated because you could think about it as an ethical way to challenge your vaccine without having someone have to like intentionally cough in your face. And like Dr. Mathee was previously saying, there is a challenge trial currently happening in, I think, the UK or Ireland, where they will be challenging directly with the SARS-CoV-2 virus, which will be a big next step in evaluating the vaccines. Next slide. Okay, so Here, we're looking at the efficacy of Moderna by race. And as I said before, the groups were predominantly white, which lowers the broader applicability of the results, but nonetheless, there were still good representations in some groups. For instance, we see in African American and Hispanic groups a pretty good showing for number of participants and 100% efficacy. We also see this in Asians. However, the number of participants was definitely lacking. And to point out the efficacy in whites being only 93.8% is because those in the study who contracted COVID were all white. However, that was only a handful of people out of 22,000. And now on Pfizer's numbers, it's pretty similar across the board with a larger population in general. However, as noted in the bottom right in red, there was a noticeably low efficacy in Asians. And this is exactly why it's important to have high numbers, diverse populations, multiple studies. We may not have seen this if we didn't have enough participants from a certain area with a certain genetic background. So further studies with more diverse populations will definitely be helpful in evolving the vaccine. And next slide. Okay, so even though we don't have a vaccine and we do have a vaccine and we've done the science, there's definitely an obvious need for the vaccine. We still have some hesitancy and resistance on many fronts, and Dr. Mathee will now go into those.
So, since we do not have the whole information on COVID-19 vaccine, I just want to use this as an example to tell you about the benefits of a vaccine. If we had all of us taken the flu vaccine in the 2018-19, it could have prevented 35.5 million illnesses, about 16.5 million medical visits, about 490,000 hospitalization. Importantly, it could have prevented 34,200 deaths. So the moral of the story, get your flu vaccine as soon as possible. And it's also important to notice that there are, about 100 million people are pushed into poverty every year globally. And it is estimated that vaccines can prevent 24 million people from entering the poverty. If this does not ring an alarm, think about what's happening in the US. 8 million Americans slipped into poverty during the COVID-19. That is like almost having one in 4 people are going hungry or no money. And it's really very sad in a country like US. Even though vaccine has such a high protective value, but there is many diseases we don't have vaccines. And there are also insufficient vaccines because countries can't afford to buy those vaccines, many countries. And to top it all, there are people who are very hesitant about taking the vaccine, as Dean Dimitrov. So to understand why there is not enough vaccines or insufficient vaccines, you need to understand the vaccine politics. So as I told you, there are many diseases can be prevented, but most of these diseases occur in low and middle income countries. And that obviously doesn't include the Western world and the Americas. And it costs more than $1 billion to develop a vaccine and takes many, many years, up to 10 plus years. So it is really not in the interest of pharmaceutical companies to also create a vaccine when you can buy antibiotics every day versus getting vaccination one day. And so that is another reason. So what changed with COVID-19? So to understand that, you really need to see this map. This is the latest map. You can see the countries where the burden is very high, the COVID-19 burden is very high and growing, whereas many of these low and middle income countries, the burden is not high and they're not even growing. And then if you look at the number of deaths, as of today, US had 315,000 deaths already reported. And you can see that many of these deaths are occurring in the Western world. But if you look at, is it proportion to the population? And of course it is not. And you can see in this slide that you have the proportionately high deaths in the Americas, and then of course the European countries, which means now there is incentive for these countries to put in the money required to develop a vaccine. US alone has spent about $10+ billion, and Pfizer got 1.95 billion of that 10, and Moderna got 1.5 billion. As you know, that Moderna was also supported by other people, including Dolly Parton. That was a big news. And now we have 2 vaccines, yet there is a lot of hesitancy. People say, oh, it's not safe. The vaccines are rushed. As you heard already today, that it's not, it may seem rushed, but we were able to do a lot of things simultaneously because we had the funding, and we did not cut corners, that were very much controlled. And then they say the body can cope without vaccine. If it can, why are we having 320,000 deaths, right? And then some argue that, oh, it will give you COVID. It cannot give you COVID because we don't have the whole virus. It's not an attenuated or killed, but we're actually giving a very small part of the genome as an mRNA. And there's a story about how these vaccines have been chipped. That is actually complete baloney, and they are not chipped. Then there's a concern about how this mRNA, this is a nucleic acid, it will tweak your DNA. Remember, this is a messenger RNA, which means it doesn't enter the nucleus, and it stays in the cytoplasm long enough to allow our translational machinery to translate those mRNA into the proteins when need the vaccine to be developed. So then there's argument side effects are worse than COVID-19. I hope Kevin convinced you they are not. And I think that is much better than having the vaccine. So certainly that is not even something you even want to discuss. And then there's an argument that make women sore. I really don't know how this can be done, so I'm not even going to try and defend that. And then there is this vaccine cause autism and diabetes. The vaccine causing autism has been, been propagated in that really truly fake news. And it has been debunked over and over with many, many experiments. I can send you a link with all the experiments they've done to show that vaccine does not cause any autism. So simply the COVID-19 vaccines are safe and effective. They prevent deadly illness. They provide better immunity than natural infections. If there's no vaccine, deaths will continue to increase. And you have to make a choice. The choice is yes. You cannot be with your loved ones in their last moment if you have a COVID-19. So you really have to consider efficacy versus the risk to decide whether you want to protect yourself, protect your family, protect your loved ones. With that, we'll end the part of the presentation and turn to panel discussion. I'm sorry, I think we went a little over. So we will now answer questions to anyone who have.
Dr. Baden, would you like to share your screen?
Yeah, I'm happy to share. This was terrific presentations. So I'm happy to share just a couple of slides.
Sure.
Because I guess I have an advantage. Let me see, share. I'm the overall PI of the study, of the Moderna study for the NIH.
Yep.
So I have a lot of inside knowledge. And so this schema here just shows you the timeline that has been outlined by my colleagues. Where from the
Pardon, can you make it a slideshow, please?
No, this is, I know how to do it quickly with the way that I know. So
Okay.
Fortunately, this I can do quickly given time.
Sure.
Is that the January 13th, we know the sequence, March 16th, first human dosed. It's then May 29th, phase 2, first human dosed. July 27th, phase 3, first human dosed. October 22nd, enrollment is complete. A month later, second vaccination completed, EUA, and we know the last 72 hours. Then the issue here, which is, again, I'm biased. This is just a thing of beauty. This is from the VRPAC meeting last Thursday. The work is under peer review, hopefully will be published in a matter of days. But this is in the public, these data are in the public domain. This is the MITT The protocol endpoint was the per protocol, which is you get both doses, you didn't have SARS-CoV-2, you make it to day 42, you then develop a symptom of COVID, get tested, and have COVID disease. Not asymptomatic infection, not transmissibility. Day 0 is first injection, day 28, second injection, day 42 here is where the per protocol set begins. So this is a secondary outcome, the MITT, but it just shows you, and these data and the Pfizer data can be overlaying each other in my view. The data are just so similar. And so you start seeing separation of curves here before second injection, but we cannot draw conclusions that one dose is protective because 97% of people received second injection. So everything after this time point is really a 2 injection time point, but then you just see the separation of curves. And then 2 key, and I recommend you all look at the VRPAC meeting, but key questions I've been bombarded with is what to do about people who are SARS-CoV positive at baseline. There were about 600 of them. There was 1 infection in the placebo group, 0 in the vaccine group. So too few to know what to do, no safety signal. Efficacy, you can infer what you want. These are the data. And then this table down here are severe disease, which is how many people had hypoxemia, hospitalized, ICU, death. 30 in the placebo group, 0 in the vaccine group. Not strong enough to say absolute, but boy, the data look in a way that make me happy as a clinician, not wanting to go round on my colleagues or loved ones or community members in the hospital. And then lastly is just the issue of when did things turn positive at the different time points. But I wanted to show you the right, I gave grand rounds this morning here. So these are my grand round slides from a local talk that I'm plucking for you. At day 28, when we gave them the second vaccination, we did a protocol-driven swab. So asymptomatic, no symptoms, they're getting second vaccination. 38 of the placebos were PCR positive, 14 of the vaccinees, suggesting that there might be a decrement in PCR positivity in an asymptomatic format in vaccine recipients. Not strong enough to declare policy, but encouraging data. So I just wanted to build on my colleagues' talks because these data are from Thursday. They were presented to the FDA. I welcome the VRPAC books. Hopefully will be in a peer-reviewed format in a matter of days so we can all discuss it more vigorously. But very encouraging, I think, both vaccine, the mRNA platform, the spike protein, and the evidence of preventing the occurrence of symptomatic COVID illness, which is the way both studies were designed.
Thank you, Dr. Baden. That was fantastic. Very good supporting data, what we have just presented.
So I'd like to open up for questions. We do have a few minutes. I know questions have been answered in the chat, but I again want to encourage everyone at this moment, if you'd like to ask a question, please proceed.
Dr. Dimitrov, it's me, it's Robert Saxton. I just want to ask Dr. Baden an important question because with 5 breakthrough cases, potentially, just potentially, you could have altered immunity in those 5 people that just didn't mount a significant immune response, or alternatively, there could be as many as 5 new genotypes of the virus that just happen to escape this particular construct of the spike protein. So I'd just like him to comment on that, give us some framework.
So Robert, I think that there are several issues embedded in your comment. One of the key things that we are trying to do is a correlate of protection. A correlate of protection actually requires vaccine failure. And with a vaccine that's 95% effective, we don't have that many failures, which is why pooling across studies may help us figure out the reason for failure and the correlate of protection. There's also the issue that you raised, which is why is there failure? Could the failure be because you have viral escape? There's immunologic pressure, but there has to be pressure on the reservoir to selectively favor certain viral mutants emerging that then can escape the vaccine-induced immunity. That is possible. The swabs exist, so the sequences can be looked at, and that will be very important to see if there are viral variants associated with failure. And then the other point that you're raising is maybe it has nothing to do with the virus, it has something to do with the host. And are there host factors that might be relevant? And I think all of those are critically important to be looked at. Looking for a correlate of protection is a high priority, but everything about this has been moving so fast that we're still accruing the data. You know, a key piece of data that I really want is asymptomatic infection. Have we prevented asymptomatic infection? And that is doing serologies now on the 30,000 volunteers to see how many of them had COVID infection but didn't know it. And is there a differential between vaccine and placebo? So all of these are very important questions, and hopefully over the next weeks to months, we will quickly generate some of these data to guide the next steps.
Dr. Baden, I have an economic and clinical trial question. We have public money perhaps and private money steering the clinical trial process in Pfizer and Moderna. Will that at all change the way these formulations are tested in the clinical trial realm, or is it, there shouldn't be any worry there, is that right?
Well, so your point is very well taken, which is Operation Warp Speed, which the US taxpayer has heavily funded, is funding 5 out of the 6 of these. Pfizer went on their own, so they're not part of the OWS. Moderna, Janssen, AstraZeneca, Sanofi, which one am I forgetting? Well, I'm forgetting one of them, the other protein. But the 5 out of the 6 accepted or were heavily subsidized by the taxpayer. What that did do is force a commonality of study design, of endpoints, of FDA processes, of data safety oversight, which was the NIH DSMB, as well as sample collection to look for some of the issues that Dr. Sackstein raised, is that we have a similarity of collection of samples so we can ask these kinds of questions. So in that sense, I think it has enhanced the science, hopefully enhanced the public trust, and hopefully has enabled us to have avenues of investigation that will make sense to us. In terms of what it will do with the rollout is going to be by the public health authorities. I think Pfizer and Moderna both turned it over to the CDC / the states. And so I know in Massachusetts, rollout is determined by DPH. And I think in Florida, your DPH as well is guiding the rollout. I assume most of our state DPHs will follow the CDC or ACIP recs with some modification relevant to our locales.
Dr. Baden, I have a quick question. So now that the phase 3 trial is over, we know which are the people who got the placebo. Are they going to get vaccinated?
So I think that a couple of things. Now, the issue of what to do with the placebo group was a hot topic at the FDA meeting and is still a hot topic for a variety of reasons. I would argue the study is not over.
Yes.
The study is not over. The study is not over. We have 30,000 volunteers in the study. They are 6 months ahead of the rest of us in terms of getting vaccinated. What's the durability of immunity? There are a variety of questions about augmented risk a year from now because of the theoretical enhanced disease versus there is no change in protection. And this group of 30,000 individuals are on the vanguard. We still have to learn about the asymptomatic issue. We need to nasal swab them at the end of the double blind part because maybe we can double our numbers about carriage in the nose that could influence the assessment of transmissibility or potential infectiousness. So I disagree with some of my colleagues in other forums where we should now end the study and treat them as if they were community dwellers. They are 30,000 people in a high quality RCT that has to change for ethical reasons because 2 EUAs have been approved and many of them are at very high risk for acquisition of SARS-CoV and for severe illness. And therefore we have to transition them in a way that makes sense, that allows them to stay in the study so we can keep learning. And this vanguard can continue to inform us that a year from now, is the protection as good as it is now? The protection that we know now is short term. It's 2 to 4 months post-vaccination. Yeah. I think it's going to be good for a year. but I wouldn't bet on that. I don't know. I want data. The only data we're going to get is from this group, or we turn it over to observational data, which we will accrue as well. But observational data, in my view, are never as strong as RCT-based data. So I don't know if that helps answer the question.
No, that's an excellent answer. But unless the placebo group, they're willing to stay on as a placebo, then we are morally committed to vaccinate them.
Well, let me clarify. I would cross them all over to open label vaccine. So now we have early versus late vaccine. I apologize if I came across that they should stay as placebos. No, there are 2 EUAs that say they should be vaccinated. In the Moderna study, 25% are healthcare workers. Another 17% have high-risk features for severe morbidity. Another 20-30% are people who are in the 1B, first line. So we're talking the majority of the volunteers in the study are at high risk. And so I think they should all be crossed over in an open labeled manner and then followed as early versus late vaccine recipients. And that differential can then be followed through time to look to see if there's any erosion in the efficacy or any safety signal that might be related to early versus late vaccination. I don't see how we can justify maintaining them in a placebo group when we have anywhere from 2 to 5 severe cases a week. And therefore, how many more severe cases do we need to see to convince ourselves of the efficacy? And is there a lack of understanding of the efficacy, at least in the short term, over a matter of the next 2 months? Because it's only a matter of 2 months or 3 months before everyone gets vaccinated anyway. So we're arguing over a few weeks. Right. And then all the confusion associated with 100 sites in dozens and dozens of states, all with different rules about rollout. So the confusion here has the potential to be very high and frustrate the volunteers substantially.
So I want to address 2 questions posed here. One is this new strain of virus that has shown up. Yes, some of the new strains shows a higher infectivity rate. To date, according to the vaccine manufacturers, it doesn't prevent the vaccine from being effective against the strain. So we are still very good on that front. And maybe Dr. Baden can add to that. Do you have any other information?
Well, I know the D614G, which is certainly in this country, doesn't seem to alter the target. So I think is unlikely to be a significant issue. The strain in the UK, they're all excited about. I think we have to study it a little bit more to make sure intuitively, without selective pressure, it seems very unlikely that it would escape. But I think by looking at the sequences, that is, one can make an estimate as to the likelihood of an issue. My guess is it won't be, but again, the data.
My case is also it won't be, because I tell them, remember, they have to bind the receptor protein, which is not mutating at any time soon. So the spike protein that makes the contact is going to, even if there is mutation, it's still going to have to bind to the spike protein. If the antibody blocks this binding, it's going to be protective. Unless the mutation changes so much that binding is elsewhere, I don't expect...
The mutation they're talking about in England has several different components to it, and they're all involving different parts of the spike. Yeah. And as we all know, right, once you start modifying those amino acids, you can get different 3-dimensional conformations that can really have an impact, even if it isn't directly at the receptor binding domain.
And one other point here
that still remains to be seen.
One point here that has to be made is in one of the viral phenotypes, it's an asparagine to a tyrosine, which means you could actually be morphing a glycosylation site, and that becomes a little bit more problematic than just API. The other point is, if I heard correctly then, the Pfizer vaccine is the monomer form. It's the trimer form from the Moderna, which means it has to have the transmembrane region. By implication, do we know if the Pfizer vaccine is missing the transmembrane region? Because that would be critical to understand a little bit more about its efficacy down the road.
So they do have the Pfizer vaccine has a transmembrane anchor.
So that's the trimer. That is probably creating the trimer as well. Both of them are trimer spike proteins. Mm-hmm.
Yeah, they have the transmembrane anchor integrated into the mRNA, so they do go to the membrane. So the other second question I want to address is the sulfur drug question. As far as we can see, the formulations don't have any sulfur compounds. And Dr. Baden can correct me, but I think I don't see any sulfur compounds in the formulations in both Pfizer vaccine and Moderna vaccine.
Agree, shouldn't be an issue.
Dr. Baden, I have one question. You mentioned durable response. Is your clinical trial designed to assess high affinity antibody and T cell reactivity a year from now?
Humoral responses, no T cell responses in the 30,000 person study,
Okay.
just by the samples that are being collected. Although sub-studies can look at the T cell compartment, but that needs to be done in a more targeted way, given the labor in collecting information.
Yeah.
So
Okay.
far, they have been doing T cell ELI spots, Dr. Baden, because they didn't
Yes.
support the interferon gamma production.
No, that's the phase 1. So in the Lisa Jackson's work and with Barney Graham, they have looked at that in the phase 1 work. So the data do exist in a limited, focused way associated with the phase 1 studies, but not in the phase 3 study.
Yeah, I mean, I think that I will also add to that some of the markers were not done with every patient, like especially Pfizer didn't do all the markers testing. They do test the select groups for select properties or select markers. So yeah. I mean, like, you know, it's like Dr. Baden said, it's very early in the process. We just finished the phase 3 and we have to do more, and we have to keep monitoring this first group that is already received the vaccine. And in a year from now or 6 months from now, there should be another set of analysis that will come out, and it will continue to come out for next couple of years before we say, yes, we have it, we got it. This is the vaccine. And then, of course, as the virus changes, we're going to keep tracking how good is the vaccine against these new variants, and that gives the companies more incentive to continue to improve or create new vaccines, just like the way we do with the flu vaccine. We make a new vaccine every year, and we will continue to create new vaccines.
Okay. Maybe one more question. Time for one more question. I just want to remind everyone that this session was taped and recorded, so just email me if you'd like a copy of this presentation in video form. This has been an incredible hour and 15 minutes. You know, the amount of participants that were involved, the knowledge and information that were shared. I feel pretty lucky right now to have been through this session. Let's hope that we can continue these types of discussions in the future. One announcement before closing: this journal club is going to be turned over to our HWCOM medical students, and they're going to run the show from here going forward. I'm involved in discussions with the second-year students to help organize this journal club. Again, it's a big success here at the college, and I appreciate Dr. Mathee, Dr. Baden, Dr. Raymond, and Kevin. Excellent presentations. And you know, I hope everyone has a chance to read a little bit more about the vaccines, and we'll line up at when they first become available, and let's all become immune to this pandemic. So it's been a hell of an afternoon. I again appreciate the attendance, and hope everyone stays safe and have a great holiday season.
I also thank Dr. Dean Dimitrov to organizing this and inviting me to lead this discussion. I thank Kevin and Dr. Raymond for being a member of this process, and Dr. Baden for coming and joining us today. Please do leave your questions and emails, and we will be happy to generate some really informed answers that we'll share with you in time as soon as we finish the recording. And we'll send you the answers along with it. And please stay safe. If you get a chance, get the vaccine. Don't forget the flu vaccine as well. And I wish you all a very happy holidays, and see you all in 2021 safe and healthy. Thank you.
Thank you. Take care, everyone. Bye-bye.